Functions of cell membranes include facilitating compartmentalization, providing a divide between the intra and extracellular spaces, facilitating exchange of substances, providing and maintaining electrochemical gradients between two sides of the membrane, receiving extracellular signals, and triggering subsequent intracellular responses through receptor-ligand interactions. Membranes also establish intercellular communication. The physical organization and functioning of biological membranes depends on their constituents, among them lipids, proteins, and carbohydrates. To simplify, lipids establish the physical integrity of the membrane and create barriers to the rapid passage of hydrophilic materials, such as polar molecules and ions. This is achieved by means of a lipid bilayer around 75 angstroms wide. The lipid bilayer serves as a lipid lake in which numerous other constituents float. These constituents include numerous membrane proteins, including integral membrane proteins, which are embedded in the bilayer, and peripheral membrane proteins, bound to the surface of the membrane via non-covalent interactions, ionic, hydrogen bonding, or bonding to integral proteins. Lastly, lipid-anchored proteins are covalently attached to a lipid molecule which itself is embedded into the bilayer. Many signaling proteins use this. Alike, islands floating on a lipid lake are lipid rafts. These are microdomains of the plasma membrane that are more tightly packed and less fluid than the surrounding membrane due to their high concentration of sphingolipids. Lipid rafts contain specific proteins, including signaling molecules and receptors, which are concentrated for efficient communication. They are also involved in cytoskeletal interactions, acting as anchor points for actin remodeling. The fluid mosaic molecule of the cell membrane was developed by Singer and Nicholson. Its tenets are as follows. The membrane is a fluid mosaic made of a lipid bilayer with proteins embedded. These proteins interact with each other and with lipids. Membranes are dynamic structures. They can move in the plane of the membrane and continuously reorganize to support cellular processes. And membranes are asymmetrical structures. They show specific and different distribution of proteins, lipids, and carbohydrates in each of the two monolayers. An example of membrane asymmetry is that carbohydrates in glycoproteins and glycolipids are only present on the extracellular side of the plasma membrane. Additionally, peripheral proteins are bound to one side or the other, and lipid composition is different in each layer of the lipid bilayer. For example, phosphatidylserine is found in far greater quantity on the inner leaflet. The membrane's asymmetry can be observed by means of cryofracture. Cryofracture is an electron microscopy preparation technique in which the sample is rapidly frozen to immobilize its structures, and then the sample is fractured along planes of natural weakness. The lipid bilayer is held weakly together in the middle, its hydrophobic core, so it splits into two leaflets. After fracturing, exposed surfaces are shadowed with a thin layer of metal, producing a replica that can be seen with electron microscopy.